Good evening, everyone. Welcome to NV's webinar series on life program. So, IISc-EIACP Center, along with Soaring Sparrows Foundation, handling the series of webinar under the life theme. So, life theme is a unique initiative of Prime Minister Narendra Modi ji. So, life was started by Prime Minister Narendra Modi ji at Glasgow in COP26 on 1st November 2021, which is a mass movement for mindful deliberation and utilization instead of mindless destruction and consumption to protect and preserve the environment. So the vision of life is for all humans to live lifestyle that is tuned with the planet, does not harm the nature. Those who live the lifestyle is known as a pro-planet people, and Mission Life borrows the past and operates the present and focus on the future. So there is a imminent threat of climate change, and because of that, there is a threat to biodiversity, but also the direct consequence of mass extinction. Along with that, there is a continuous pressure on resource usage, and also because of this unethical resource usage leading to the destruction of the planet. So, in this regard, Life program is aiming to target to protect the environment, and also it is considered as one of the nationally determined goal to contribute to the healthy and as well as sustainable lifestyle. So, in this regard, the EICP Center at University of Science, we are conducting series of webinars, and today we have with us Dr. Karthik Bala from Aquatic Institute. So, Dr. Karthik is a scientist, and also he's leading the biodiversity and radiobiology group. So he is working on diatoms, and his lab working on study of living as well as fossil diatoms, and he has extensively worked on the various terrains. He has traveled across the Western Ghats as well as other parts of India, and currently is working on biomonitoring of the lakes, rivers, biofuels, and forensic. And he maintains the South Asia's most expensive diatom collections at the Agartala Research Institute, Pune. And his research institute interest includes taxonomy, ecology, and biogeography of the extinct freshwater diatoms, and also understanding the application of diatoms in the environmental pollution monitoring. And also he is working on diatom-based environmental reconstruction, and also he is working on biofuel and also carbon sequestration and nutraceutical applications. And he has working in Agartala institution under as a scientist from January 2022 till date, and he was worked as a scientist in the same institute from 2018 to 2021, and also he has worked as a scientist in from 2013 to 2017. He was a postdoctoral fellow at the Department of Environmental Sciences and Management, Northwest University, South Africa. So he was a postdoctoral fellow at the Museum of Natural History, University of Colorado, USA. He was a research associate at Center for Ecological Sciences, Bangalore from April 2021 to January 2012. He was the earlier postdoctoral fellow at the Museum of Natural History, University of Colorado from 2010 to 2011. And he was worked as a research assistant at Center for Ecological Sciences from 2004 to 2010. And he has done his PhD from University of Mysore and MSc from Bharathidasan University and Bachelor of Science from Periyar University. And he has been awarded T.N. Kushur Memorial Award in the year 2022 for biodiversity exploration, organized by A3 Bangalore. And he has been serving as a member for the various national committee as well as academic service. So, worth mentioning that member as an expert committee under DST for the Jawaharlal Nehru Center for Advanced Scientific Research. He is also a selected member for the 4th Indian Bioscience Outreach Grant Program coordinated by India Bioscience. He is also a member of Year of Awareness Science and Health Program by the DST, and he is the associate editor for the Diatom Research from 2022 till date. So, with this brief introduction, on behalf of EIACP, I wholeheartedly welcome Dr. Karthik for delivering his lecture.
Hi Bharat, can you hear me?
Yes, I can hear.
Okay, thank you for this kind introduction. I think we are already late by 5 minutes, so I'll start my lecture immediately. So the topic which we are going to discuss on role of diatoms in securing energy for future. So in case if you lose me in between, just tell me, then I can go back and then repeat those slides anywhere.
Sure,
So
certainly.
yeah, so diatoms are microorganism with macro function. If you see that, this purposefully these two fonts are at um, a various size, just because the microorganism which we need microscope to see, and macro function because they have contributing almost 25% of global oxygen. So even though we don't know about this a particular group of organisms that much, but they have play a very critical role in ecosystem functioning, and their service to ecosystem is huge. So the flow of today's talk is going to be, I will introduce diatoms. I assume most of the audience will not know much about diatoms, so I'm going to introduce diatom, then talk about diatom biofuel, and then value-added products from diatoms. So when we started looking at diatom in 1703, if you see this person called Mr. C, he observed this uh, uh, green scum material from his pond, and then he documented this morphological feature which you see on the left. So and then he communicated to Philosophical Transaction of Royal Society of London that he saw this kind of observation. He don't know that time the item itself was not very well known. So this is the first ever documented diatoms. And then almost after 150 years, this gentleman which we are seeing on the right, Edinburgh got samples from some of the visitors who visited India, various parts of India from Europe. So that time they sent samples to him. Then Edinburgh was one of the most celebrated microgeologists, because then the term microbiology was not there. So it's like microgeologist. So he used to describe diatom, coccolithifers, and foraminifera, like various group of organisms. So uh, just a minute, hold on. Sorry, the door got locked. Okay, so if you see these highlighted places in this right side document, you see places which is very familiar to us, like Western Ghats, Nilgiri, Srirangpatnam, Malabar, Ganges, Brahmaputra, and Deccan, Pondicherry, Tranquebar, and Mangalore, and Canara West Coast. So you see this Canara West Coast. So these are the terms which are very well known to us. So diatom was described from these places in 1854. So most probably India's first observed microorganisms are diatom, because I don't think anybody else described any microorganism before 1854. So this must be the first described microorganism in India. So here is a very celebrated Victorian art. So each in this owl, or in this arrangement, like something like Rangoli. So you see each one of them, each one of this is a individual diatom. So it is arranged meticulously under the microscope, and it is arranged in this kind of slide. This is a very expensive slide, and in Victorian time, this is very important thing to possess. So this slide, if you observe, you can see this kind of pattern. So many designs in European culture inspired from diatom. So these kind of designs you can see in the wall or in the ceiling. So what are diatoms? As the names suggest here, it's single cellular. It's a single cellular golden brown algae, because apart from photosynthetic pigment, which we know clearly, like chlorophyll. Bharat? Hey, Bharat?
Yes, please tell me.
Yeah. Is it okay to ask audience two questions, like how it's like nature of the audience? Or like,
Generally, we take it in the end of the
no,
presentation. If you have any specific question, we can ask us.
no, I can ask audience.
You
Can I?
can pose the question. If somebody want to share their views, they can do that.
Sure.
Generally,
Okay.
we have it in the end. We have it in the end.
Okay. If anybody have any question, please unmute yourself, and then, because in the online mode we don't see the participants, so we don't know whether somebody is there or not. So it's always better to keep it in, interactive from both the side. So anybody have any question, please stop me at any time, and then you can ask me questions. I am happy to answer. Or like if you have any comments,
Sure.
also we can discuss over it. So
Sure.
what is
The chat is
specific?
also enabled. Chat
Yeah.
is also enabled, so they can also type in the chat.
Sure.
Sure.
So, one of the very unique feature which you see about Diatom is that what you are seeing here. This is a siliceous cell wall, and the siliceous cell wall have a pattern that these patterns are unique to each species. So we use these patterns for identifying one species from other. So whatever you see these dots in this area, which you are highlighting, so this is called areolae. So these areolae patterns are unique to every species. There are 60,000 plus known species are there. So everything have its own. We use a combination of this entire wall morphology and these character, and then multiple characters we use to identify the species. So diatoms can live as a single cell, as you see in the pictures, or they can be also colony, and they reproduce very rapidly. If you put a culture in a day, you will get like 1 liter or 2 liter of culture, and they are found in any wet place. We don't need to get a really wet place, like for example, like river, lake, or stream, marine, or ocean environment. Even there are little moisture is there. Like for example, if you have a small pot in your balcony or in your window, so even you pour water there, so even that soil also contain uh, diatoms. So something very specific to diatom is there are environmental specific. Diatom which you see in saline and in saline, in seawater, is different from fresh water. In fresh water also you will see like lake diatom have a different set of diatom, and then river diatom are different. So this is what you see. So based on looking at the diatom, we can tell, okay, this is river diatom. In river or in lake, you can see it is attached to stone. There are specific set of diatom. Attached to plants, there are specific diatom. And in stone and plant also, we can see polluted diatom which can tolerate pollution, and then diatom which are sensitive to pollution. So we can see all these kind of differentiation among diatom. So there are 50,000 known diatom, but this number is increasing extremely drastically. Every day there are hundreds of species getting described. So, and the total estimated is 2 million diatoms, which we know only 50,000, and the total estimated is 2 million. So you can see how diverse and how we know less about diatoms. So how we can see diatoms through our naked eye? You can see here the stones attached, covered with the biofilm. This is algal growth. Diatom also major part of it. This is a stone, and here you can see these plants filled with diatoms. And then sediments, you can see the sediments with the golden brownish color here. And then there are on the moss, and then you can see this seepage will be filled with a biofilm which is dominated by diatom. And then they are also occurring as a plankton, like free living in the plankton in a lake or river water. So this of diatom looks like, you can see the silica cell wall here. This is silica cell wall, and then these are chloroplast. So that's how they photosynthesis. You can see various shapes and size of diatom. So how do we study diatom? So we collect, like as I showed in the previous, like if you take this stone, we using a clean toothbrush or a small knife, we remove this biofilm carefully. Like if you see here, or in this portion, you see this plant diatoms are like brown growth attached to the plants or the sediments, and then we remove them from that original area habitat, and then collect it in the small polythene bag. And then we are interested in looking at the silica cell wall, like this silica cell wall. So we boil with nitric acid or like any strong acid, and then we look at the diatom in this is light microscope, which is 10 it's like 1000 time magnified, and the bottom one which you are seeing is scanning electron microscope. We can go up to 50,000 times we can magnify this. So this is how diatom look in the light microscope. For any ecological assessment, we do community enumeration. So we count individual diatom, like this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Like that, we do 600 diatom per sample. So that's how we study that community like diatom assemblages. And while doing it, we also look the diatom under the inverted microscope, where you see the light sources coming from top, and then so you get this working environment where we pick single diatom. We pick a single diatom, and then we understand their chloroplast morphology. We pick the single diatom and we grow it. We do a culturing. So here you are seeing the same diatom to understand the chloroplast morphology, and this is after acid treatment. So now here you can see these characters very in detail, all this striation, areolae, which I was mentioning a couple of slides before. So you can see all this thing in detail. So further, we do in the scanning electron microscope. So you can see the same areolae is in detail. Like we can see this is like, it's all arranged in nanometer level. So where we study diatoms, these are the places where our lab have collections. So we have more than 4,000 samples from various parts of India, but our major focus is on Western Ghats and then Northeast India, because these are the two biodiversity hotspot. So we focus most of our study in this region. However, we also have samples from Western Himalayas and Andaman and so on, so other places as well. So diatoms can be termed as a life in extreme. For example, like across all parameter, like if you take, for example, like you heard about this lunar crater. It's like a soda lake. Why we call it soda lake? It's the pH is more than 9 pH. On the other hand, you have Meristica swamps. For EWRG member, Meristica swamp is they are very aware of it, because today all over the world people know Meristica swamp, particularly from the work of EWRG group, because I can proudly say I am the proud member of this group. So where this habitat has went to become very famous, studying various life forms and its conservation and so on. So this Meristica swamp, one of the characters is acidic pH. You can see the pH is less. Sometimes it can go to less than 5. In both the environment, you will find diatoms which are new. So either in Crater Lake also you will find diatoms which are new, and also in the Meristica swamp also you will find diatom which are new to this environment. Likewise, you can also find diatoms in a clean environment like this lake on 2,500 meter above mean sea level, or like you can get like this dirty place where we are seeing in the middle of the Pune city. There also you will find diatom. So in this process, we describe multiple new species of diatom from various parts of the country, and even new genus also we have described. So this is about the basic idea about diatoms. Now we'll move on to the today's topic, which is on energy related topic. So diatom for biofuel. So diatoms are in the radar of biofuel applications since 80s. So that comes from this famous program called Aquatic Species Program of Department of Energy of USA, where they screened various groups of algae for the potential as a biofuel, and they screened around 3,000 species of various groups of algae, including diatom, which also the algae sourced from freshwater, brackish, and saline water, and they assess what are the potential of various groups of algae, and they came up with this system called open pond system, where you have, this is a circular pond where there is a motorized pedal is there. So this pedal will push the water, and there will be also CO2 will be pumped into this, and then we'll add nutrients, and then there will be a circular movement, and then there is a growth, and at some point of time this algae growing here will be harvested. So in this study, that this is from the aquatic species program, you can see, like, in 1980s they started, and you can see that what they looked into various aspects of algal biology, starting from taxonomy to physiology, and then lipid biology, and then even at the genetic engineering of algae for higher lipid production. And then that the first one was done with the first successful genetic transformation done using a diatom itself. So why we need to go for algae? There are three class groups you see here. These blues are commercial, and then this green are demonstrated, and then this red one is targeted. So if you see wherever algae, it's way ahead of all our other commercially used biofuels like soybean or jatropha or ethanol, everything we can see. So that's the target advantage we have using algae. And apart from that, it have a high economical, environmental, and nutrition advantage. In country like India, where we have more than a we have a huge population, where we cannot compete with the agriculture, so we cannot take away the food crop and then put a biofuel crop. So we need to find out a way where there is another alternative where we don't touch the food crops. So in considering all this, when we select a best biofuel, it should meet all this criteria of economic, and then food, and then environmental. So for that, like, algae is one of the best candidate. So in terms of when we use algae, it produces almost 40 times more protein per hectare when compared to the all other alternatives, and then 20 tons of oil per hectare when compared to the most commonly used palm, which is like hardly 3.3 ton. And then there is not there is no evident pollutant coming out of the algal farm, rather it sequesters carbon. So in that way, the algal farm are way and more environmental friendly when compared to the other alternative. Apart from oil, we will also get protein, carbohydrate, fuel, feed, and other polymers which have high commercial value. So there are some statistics which says 30 algal farm would restore the entire ocean ecosystem. And for example, the 40 algal farm can compensate the entire deforested area of Amazon forest. So like 900 algal farm can restore the entire Southeast Asian rainforest, which are removed for the palm oil. So, and 1,500 algal farm would reduce the a drastic amount of greenhouse gas emissions. So like that, we can keep on going on this number. So that why diatoms are one of the very celebrated candidate for energy, because they make huge amount of lipid on their system, particularly when they are stressed. So I just want to share with some small case study that we collected samples from various parts of the country, and we brought it to the lab, and we picked single cell, and then we established culture. Like this is how the culture looks like. This is like similar to tissue culture facility, and then we characterize this diatom using morphology and molecular method, and then we characterize the lipid content by gas chromatography. So this is a drop, like not even a small drop of diatom from the culture, so you can see how densely they grow. So for example, like now I just want to demonstrate with one particular species. So like the species called Gomphonia parvulum. This species occurs in any water. Like even if you take a sample from a extremely polluted sewage, or a lake or a river, you will find this diatom. So why we choose this diatom is because we don't want to provide a special nutrient to grow diatom. It should be able to grow in a from a sewage. So we can also do nutrient recovery. So what sewage already having high amount of nutrients in terms of nitrate and phosphates. So by growing this, we can recover the nutrient, and then we can also get the diatom to produce lipids. So this is how they look in the live diatom. So you can see this live diatom, and once we do acid cleaning, so we can see how they look like. So this is for just for taxonomic confirmation. You don't have to do these steps when you are aiming at oil production. This is how they look at in the scanning electron microscope. So we also to confirm their identity, we also do molecular identification to find out what species they are. So so these red dots are the lipids. We have markers which will highlight the lipid. So this is the control, and on your left you are seeing control, and then this is media without nitrogen, and then media without phosphates, media without silica, and then media without phosphates, nitrates, and silica. So you can see this is 6th day, and this is 6th day, and then this is on the 12th day. So you can see the one which is in a phosphate deprived condition produce high amount of lipid. So this is the kind of it's almost kind of saturated. So this is a kind of stress which will enhance the lipid growth. So if you look here, so this is various on the x-axis we are seeing various cultures, and then their dry biomass weight, it goes up to 50%, and then you have the lipid weight only, which can go in some cases it can go up to 50, and then it typically range from 25 to 50. This is the range. And some of those we do GCMS analysis. You can see so many unique fatty acids associated with these diatoms. So our library produced when we whatever the results which we get, when we compare, we get a wide array of fatty acids. We get it from the diatoms. So like this, we also standardize what would be the growth curve, how fast they can go. Typically, if you see all diatom reach their maximum by 10th or 11th day, you will see the maximum growth occur. So for some species, it happens in 9th day. So this has been then characterized for all species which have high potential. And so this is done from our lab. So now whatever I'm going to show, it's all from the published case study. So these, apart from this, there are so many studies which have particularly many reviews. If you published reviews which shows that what are the prospects diatom have in commercially biofuel application. So while we grow diatom for the biofuels, and we also find other value-added products from diatom. This is very, very important, because diatom, it's not only for biofuel production. For example, if you look at any other oil crop like palm or like jatropha or any other crop, so they are cultivated mainly for the oil production. But in terms of diatoms, you get they have a wide range of product. We'll go some. So as I mentioned, they can be used as a nutrient removal from wastewater. So while we growing diatom, we can also grow. We can also remove the pollutant. Particularly, diatom need N, P, and silica. So this N and P is the most important cause for the eutrophication, so that can be removed, particularly with respect to the uh, non-toxic organic sewage, and then like sewage from the uh, fishery waste, and all those things can be uh, removed using diatom. And uh, if you recollect, I mentioned something called areolae, the term called areolae, which is the these holes which are on the diatom surface. So if you zoom in, like this is like 500 nanometer scale. So you can see there are so intricately made patterns are there, and these are used as a template to make some nanostructures, which is used in solar cell and then lithium ion battery and many like capacitors and photonic crystals, and also as a drug delivery vehicle in various applications. So diatoms are already naturally made nanostructures. So these nanostructures are used to replicate such structures, which is having multiple application across the field. Silica also so once we grow diatoms, as I mentioned, diatom are made up of silica cell wall, right? So the silica cell wall, once we extract the lipid or organic part of the diatom, so we get a huge siliceous deposit. So previously people avoided using diatom because it make huge silica debris kind of thing. So what to do is like, but now people have found it can be used as a material for in place of lithium solar cell battery. In our lab also we are trying to do like assessing the storage capacity of diatom derived silica, and they're also used in the bone engineering. So diatoms are used as a in case of where there is a damage, so the fillers can be made using the diatom cell wall, and they are also used in as a hemostatic agent in the to control the blood loss in like there are this chitosan is one of the very well known nanoparticle which is used for to control the bleeding, so that it is mixed with diatom, and that's used as a one of the most commonly used hemorrhage control material. So when we are looking at it, so we are also this is the era where we're talking about CO2 sequestration. So when we talk about CO2 sequestration, we are all thinking about uh, growing trees, social forestry, and all those things, which takes a lot of time and effort, and we need to convert a land use from agricultural to to forestry and so on. But if we leave a lake by itself, if we just maintain a lake, by default it does a lot of carbon sequestration work. And diatoms, as I mentioned, they produce 25% of uh, oxygen. So and then approximately they sequester 20% of the global carbon dioxide. So when we are getting oil, biofuel, they are also having a lot of byproduct. On top of it, they are also making sequestering a huge amount of carbon dioxide, which in turn have a lot of effect in reducing the greenhouse gas and eventually in climate change. And by doing so, we also get carbon credit, which has been recently introduced. It will soon be coming to market. Like if you grow tree, so you can get paid. Uh, if you have a land, you can if you are growing tree, and based on the amount of estimated carbon sequestration, you can get paid from the government for your effort in sequestration. So the challenges we have here is selection of species. If you recall, we have 60,000 plus species are known, and then we have more than 20 lakh species are unknown. So which one to select? Already we have very limited knowledge about our diatoms and its diversity and what kind of environment it can happen. So we don't know any of those things. So which species to select? So immediately many people who are having a commercial algal farms, they contact me and ask like, what species to use. So we are in the process of screening and coming up with some potential species which have high biofuel and other byproducts. And this is a new field, so we need a huge investments from the industrial sector to come and invest and then to uh, harvest at a large scale. Until we do a large scale harvestation at multiple places, otherwise it is difficult to find a market where you can sell. For example, on the picture which you are seeing is a spirulina uh, farm in Pondicherry. There are a lot of spirulina farm available across the east coast, particularly like in Andhra and Tamil Nadu. So you can Google spirulina farming. They teach classes online. So you earlier it was restricted only to the coast. Now it is available even in inland. So we need to uh, develop a technology which can reach a grassroots level. So even some villages with the limited uh, understanding should be able to cultivate diatoms. So that's the one of the main. Then only we will be able to improve life. Like it's not about commercially making it viable, but this has to reach grassroots level. So everybody can cultivate diatom and then sell it like that. And we don't have a designated culture collection where I can place the order, I can buy A species, B species. So that is another one drawback. So we are in the process of developing a diatom culture collection, which will be soon supplying culture to first to start with academic purpose, then later we are planning for the industrial application. Right now the market for diatom culture and diatom product is very limited in India, because globally we have handful of huge diatom based plants, where many plants in western they produce on biofuel, and some plants on Japan they produce on pigments, because diatom makes some of the very interesting pigments, including some diatom, like I can mention like some, including some diatom which make blue pigment, which have a high commercial value in, especially in the cosmetics. So at the moment we don't have a huge market, so these are the challenges which will be addressed soon. So with this I conclude my talk, and I'm ready to take your questions.
Dr. Karthik, thank you so
Bharat?
much. Thank you so much. So now I request participants can unmute themselves and ask questions. Or if you have any questions, you can specifically share in the chat box.
Sure.
Dear participants, you have any questions, please post to Dr. Karthik.
Uh,
Actually, I have received one question from YouTube live stream. So
okay.
uh, he says algal blooms can cause adverse effects and which can change the water chemistry and increases the pH and also swings in dissolved oxygen. In that scenario, whether we can introduce diatoms to reduce this algal bloom effect.
No, sometimes diatom also cause bloom. Even it had created some bloom in Arabian Sea. So diatoms are algae. There are 5, 6 different groups of algae. Diatoms are one of those algae.
Okay. So there is one question from Tulika. What are the challenges involved in the implementation of extraction of biofuel in small scale industry?
So, as I mentioned in the last slide, there are so many challenges out there. Like we don't know what species can produce how much biofuel, first thing, and then we need to have a market. So, if I take it to introduce it in, it should be brought into something like like growing a goat, or like we have some crop at home. So uh, where is the market, and there should be a plant where the item which is bought from somebody can be processed, and like that there are so many challenges are the industrial level challenge.
Again, I have got one more question from the YouTube. So,
Yes.
diatoms do not decay easily because of having siliceous walls. So, in that scenario, how these diatoms can decay?
So diatom will not decay. Diatom, the cell wall of diatom is made up of silica, which is inert material. Okay, so this the cell wall will remain forever. To give another example, like there is a material called diatomite. So diatomite are the dead diatom, which is from few million years ago. So there are dead silica. The once the diatom die, so then the cell wall settle down. Under the in the marine condition, there are mountains of dead diatoms are there. So the silica cell wall remain there forever.
Okay. So, is there any questions, participants? Is there any questions?
Is there enough manufacturing in there is a
Oh, there is a
Tulika,
one.
is there is enough manufacturing infrastructure in India for extracting? Uh, right now it is not there, but we can it can be made because the technology is very similar what we have for extracting of oil, esterification, and all those things. So it's possible to make.
So,
So there is one more question. Okay, thank you. Okay.
okay, I'm just looking at other places also. So there is one question. So have you noticed any morphological changes between marine and freshwater diatoms? If yes, can you highlight the change?
So the marine diatom don't occur in freshwater, and freshwater diatom don't occur in, because they are very specific to their environment. But marine environment is predominantly dominated.
Hello, Dr. Karthik, we lost you. Hello, Dr. Karthik, we lost you. Hello, hello.
Bharat, you have to call him in mobile.
Okay, okay. Hello.
Hey, am I
Yes,
audible?
please. Yeah, you are audible. Welcome
Yeah.
back.
Sorry, the power went and came, so there is a slight.
Okay. So the question is regarding the morphological changes between the river and freshwater diatom. In between we lost the connectivity. Is there any noticeable change between freshwater as well as
No,
marine diatoms?
as I mentioned, there is a drastic difference between the species occurring in marine and then species occurring in the freshwater. These two are two different set of species. We don't find species which occurring in freshwater also occur in brackish or marine environment. These are two different set of species. There is no special adaptation of same species in marine environment and freshwater or nothing like that. What we find in freshwater is limited only to freshwater. So there is nothing like that. And there was another question on how beneficial is to harvest in Western Himalayan region, because the challenge of market is also there. What is your view? And also in hilly area, can we harvest huge amount of diatom because of freshwater stream and water bodies are more dominant? Yeah, irrespective of any place in India, it's in wherever there is enough water, we can harvest diatom. But at the moment, we don't have that market, associated market. Like, for example, we need to develop some model which would be we produce milk. If we have a cow, we produce milk, and then we go and give it in a cooperative society. They take it and process it, and then whether you produce 1 liter or 100 liter, there is a place where you can go and give it like that. So that kind of facility is yet to emerge in India. There are very few plants all over the world which use diatom extensively. But I heard there are huge plants are coming somewhere in Gujarat, particularly focusing on all groups of algae. So what about environmental education through diatom? And we have a big program called Diatom Project. It's also known as Sim River. I just typing that in the chat box only. So you can Google this, and we have a multilingual environmental education program using diatoms. Today it's focusing only on energy, so I didn't talk about that. Is there any negative effect of water bodies for culturing diatoms? Yeah, it need to be a controlled environment. We cannot cultivate diatoms in a lake or a general river. We can take water from there and then we can cultivate. We cannot do it in a common environment. As such, there is no any negative effects on water bodies on cultivating diatoms.
Okay. Looks like there are no further questions. On behalf of EICP, I thank Dr. Karthik for sharing his views. Now I request Dr. TVRAMACHANDRA sir to share his remarks.
Okay, thank you, Karthik, for an interesting presentation. So,
Thank you, sir.
looks like that a lot of work is happening in your lab.
Yes, sir.
Very interesting. And recently I happened to meet somebody from Pune. They are interested in the modeling of the aquatic ecosystem and this one. I have asked them to contact you also. So
Sure.
they may contact you, because
Sure.
in their campus they have the water body and they want to have the terrestrial ecosystem. They have done the greening of the campus. With that, they wanted to do the modeling to visualize how the situation in the 10 or 20 years from now. So since you are there, instead of me traveling to Pune, I asked them to be in touch with you and discuss with you. Probably they will contact you about that. There are a group of startup, there are 3 youngsters are there in the startup. They are doing this job.
Sure, sir.
Okay, so I think we thank you for an interesting presentation, and as usual, it is quite intellectually rewarding presentation. So And
Yes,
interesting
sir.
to see that Tamil Nadu, the way the algae is grown there, Spirogyra and such things. Okay, that's interesting. Of course, I have seen this in Karnataka also. In Mysore, I have seen a couple of people
Yes, sir.
doing it, but probably that is not there on the net, I think. So,
Yes, sir. So
okay.
because this RO will have a monthly training program, so they have, and they also, if somebody is cultivating spirulina, and they are ready to take it, so the market also is there. So villagers are very, they don't have to go anywhere. You just remove the water and pack it, and then send it to Pondicherry. They have a plant of processing for tablets and then drinks and all using spirulina. That
But
plays a major.
spirulina grows in a very nutrient-rich environment, no?
Yes, sir. Yes, sir. So most of them they take brackish water from the nearby Assuran area. Now they also started, like, they add like urea and all artificial nutrition to grow in the inland part.
Yeah, I think that's what I noticed in Mysore,
Mm.
where they are growing. I think it is for industrial purpose. The
Absolutely.
protein X or whatever that this and they are
Yeah.
manufacturing that.
Single cell protein they are extracting from that.
Yeah, that's correct. Sure.
Yes.
Okay, thank you, Karthik. So,
Thank you,
Bharat.
sir. Thanks for the opportunity.
Sure. Yeah.
Thanks, Bharat.
Thank you,
So,
Dr.
life is
Karthik.
meant for a youth like you.
Sorry, sir.
Life program is meant for youth like you, so that you can do the much more work and communicate to the people.
Absolutely, sir. Whenever you want me to talk, I will be happy to, sir. Any topic, I will be happy to talk, sir.
Sure. We will
Okay.
look forward for many more lectures like that. Okay, sure.
Sure, sir. Sure, sir.
Thank Thank
Okay,
you,
sir.
Dr. Karthik. On behalf of EICP and Soaring Sparrows Foundation, so we thank you for active participation, and also I thank participants for posing the right questions. So I request everyone to subscribe to our EWRG YouTube channel so that you will get notified the coming lectures as well as webinars. Thank you one and all for active participation. Thank you.
Thanks, Bharat. Thank you.